Thank you for joining our coffee break webinar. Today's topic is tracking translocation of microplastic particles. Maria Kloukinioti completed her master's degree at the University of Patras, Greece, in management of ecosystems and biological resources with a focus on ecotoxicology. In 2022, she joined Maastricht University and TNO Institute as a PhD candidate in the field of microplastics. Her research is centered on investigating the fate and effects of microplastics upon ingestion, using various in vitro and ex vivo intestinal models.
Hello everyone, I'm really glad to be part of this coffee break webinar series in order to present you a part of my PhD research. The title of my presentation today is Tracking the Translocation of Microplastic Particles through Live Imaging. First, let me give you a bit of a background on why plastic research is so important. As we all know and notice, in our everyday lives, we use a vast amount of plastic products. From the clothes that we're wearing to the packaging that we're using to store our food in, plastic use grows exponentially over the years, and it doesn't seem to stop. The problem of having so many plastic products around us is that these products, at the end of their lifespan and through constant use, start degrading and releasing smaller plastic particles called microplastics. These particles are usually smaller than 5 millimeters and they have a really complex chemical composition, but they also come in a variety of sizes and shapes. In general, we could divide microplastics in two big categories. First of all, we have primary microplastics, which are particles intentionally produced and added to products like face creams, body scrubs, so these products can have a specific composition. Or secondary microplastics, which are particles coming from the degradation of bigger plastic products that have been released in the environment and have been exposed to different weathering processes. Secondary microplastics have already been detected in different environments like aquatic and terrestrial and have also been detected in our food chain. And if you're wondering which one poses a bigger risk, then we just have to see the production numbers and we can clearly notice that secondary microplastics are way more than the primary. Lots of research has already been done on microplastics, and it has been proven that these particles exist in human samples, such as blood and placenta. In our labs, in an effort to mimic the exposure of our gut to microplastics, we are using an ex vivo system with porcine intestinal tissue. The tissue that you see in the picture on the screen is exposed to fluorescent polystyrene beads of 1 and 10 micrometers for 6 hours. Then we are trying to track uptake and translocation of the beads by these explants. In this picture, you can clearly see that the microplastics are not only laying on the surface of the tissue, but they are clearly taken up by the cells as well. However, what still remains a big challenge is to prove that these particles can actually cross the epithelium. The first problem here is the small size of the microplastics and their limited rate of translocation. So existing methods such as the use of a plate reader, which in theory can pick up the fluorescent signal emitted by the microplastics, may not be sensitive enough to pick up the weak signal. Also, in the field, there are a lot of concerns that fluorescence might leach out of the particles and end up in false positive signal. In a way to overcome all these challenges, we decided to use the Omni device from Action Biosystems. This way, we could track particles translocated in lifetime without also handling a lot our system, so minimizing also technical errors. Since in our team we had already proven uptake of microplastics from intestinal tissue, as I showed you before during the introduction, we decided to use the same model for the translocation as well. To explain a bit how the setup of the model is, let's discuss a bit the picture on the slide. As you see, we have tissue, gut tissue, that we bring in the lab and we strip. So we remove the external muscle layer and then we punch it into smaller round segments that we mount on a transwell. So at the end, we have a traditional setup of a transwell as you usually have for your cells, with an apical side, which is your exposure side, and a basolateral side, which is the receiving compartment of something that can translocate, and in our case, the microplastics. In this set of experiments, we decided to use commercially available fluorescent polystyrene beads in 3 different sizes of 50 nanometers, 1 micrometer, and 10 micrometers. And we also exposed our tissue segments in 2 different doses of 100 and 1000 micrograms per milliliter in order to explore whether the size and the dose could have an effect. So the microscope was focused on the bottom of the tissue segment, as you can see on the picture below. So the moment that a microplastic was translocated, we could pick it up. Also, in the beginning, we did some test pictures just to make sure that the microplastics were visible on a plain well plate or on the actual tissue segment, as you can see on the picture here. Through all the videos taken, we could clearly see 10 micrometer polystyrene beads passing through. I will first play a video of one insert. Here, every small light popping up in your screen is one particle translocated, but of course this is not really clear. So next to it I have screenshots of the microplastics that you can see, and with the Omni we could track different places on the same transwell. So in every time point we could see how many microplastics were passing through in different regions of the same transwell. Also, what was really interesting to see is that we could pick up some signal on the 50 nanometer polystyrene exposures. Of course, I think what we picked up is not single polystyrene bits, but aggregates of those. That's why it's easier to pick them up. But it was really nice to see particles in this small size. And probably I think through the video it's not clear to you where these particles pop up. So next slide is a static picture of these particles. So as I said, in this picture, you can see in red circles a static picture of the particles translocated in the size of 50 nanometers. And again, they're quite small, but we can prove that through the pictures it's a particle and not just background or fluorescence leaching. From this small set of experiments, what we could conclude as preliminary data is that it seems that all the 3 sizes could cross the intestinal epithelium. The translocation also seems to be size dependent, since the 50 nanometers and the 1 micrometer particles appeared sooner in our screens than the 10 micrometers, which their translocation started a bit after the 3 hours. Of course, there might be some potential underestimation of the translocation due to unspecific binding of these particles on the support filter of the tissue segments, but this is a limitation that you always have with transwell setups. As next steps in future experiments, we would like to use a bit of more realistic particles in terms of shapes, so not spherical anymore but amorphous, and test also different polymer types to compare their translocation pattern and see whether there is a difference. To conclude, I would really like to thank the whole team from TNO that helped me with this research, and the people from Maastricht University and everyone from the Momentum Consortium that made this collaboration possible and support this research.